as I said, very warm welcome to this course regarding the Julich-Brain Atlas. Today we are starting a 4 days seminar series, and that's in particular Timo Dickscheid and his team, and you have already met Susanne Wenzel, who helped organizing this. We have a very dense schedule in front of us the next days. So we start today, Timo Dickscheid and me, with a multi-level human brain atlas in EBRAINS and provide you an overview. I'm focusing more on the neuroscientific part, on the concept. Timo is more focusing on the concept behind the architecture and how do we address the neuroscientific questions. Then tomorrow, Sebastian Bludau and Kimberly Lothmann will go with you into very practical lessons, how to use the atlas, in particular the atlas viewer, to analyze the brain. And this atlas viewer is much more than a viewer that is very intuitive and that you can use. There is really a lot of things behind, and we would like to introduce it to you. Then on Wednesday, we go one step further and approach in particular those of you who are using the atlas and the data that are in the atlas a bit on a higher level, I would say, that try to use the data to bring it into reproducible workflows using the Python tools, the Zebra toolbox. And last but not least, on Thursday, Lubert Zehl was an expert in data annotation and who has developed the Open Minds format as really one key format for the neuroscience community to share, explore, and reuse data and knowledge in eBrain. So this is also more far going, and we really would like to make the point that the eBrains Atlas is not just an atlas, but it's a whole environment where you can address all kinds of research questions. So why it is so complicated, and why it's more than just an atlas, perhaps as a traditional textbook, you know. This has to do something with the feature of the human brain that is a system that is organized on multiple levels, starting with the molecular levels. Neurotransmitter receptors are molecules that are important for signal transduction between single nerve cells. And such a nerve cells is only a few micrometer tall, but its axons can be very long, up to 0.5 meter or even longer. And this type of connectivity is different in different brain regions. So we speak about the cellular architecture, and the hippocampus is one example. It's a very specific cellular architecture, and it's a few centimeters long. And of course, the hippocampus is connected to many, many brain regions. This is so important for our learning and for our memory. And when we understand how learning and memory functions, then we have to keep in mind the organization of the whole, of the entire human brain, but also that the brain is one part of a body and of human beings. And this explains one, I would say, a major challenge that we have to address in brain research. We have somehow to take into consideration these different spatial levels of organization and somehow also bring it together into a coherent framework. And this is not an easy task and has been addressed for many, many years. Zehl and Oskar Vogt, together with Corbinian and Rothmann, these were 3 German researchers who brought in this perspective to address the brain really in a more comprehensive way. And they started their brain research at the end of the 19th century in their private little apartment, where they have set up the first neurobiology lab, and this became very quickly larger, and they were then connected to the Berlin University and later were founders of a large research institute that is today belonging to the Max Planck Society. What was their starting point? Their starting point was the analysis of the myellar architecture. And myellar architecture means that the distribution density of axons connecting nerve cells is different when you are looking to different brain areas in the cerebral cortex. And here are some examples of areas coming from the auditory cortex, from the visual cortex, and others. And you see very quickly that the axons look different with respect to where the highest density is, where you see these horizontal stripes, how broad they are, and so on and so forth. So they introduced the first concept on myellar architectonic parcellation of the human brain. And this is one of the first maps. And we still have this very nice drawing of the Vogts in our institute in Düsseldorf. And you get an impression how these old cartographers worked and how they represented areas with different myellar architecture in the human brain. So later, when the Vogts got the opportunity to found a very large institute in Berlin, an institute of brain research, they have set up it in a way that is still modern and that has a concept which is still actual. So this institute was set up before the First World War, so about 1912, 14. And then it started to be implemented after the First World War in the end of the 20s. And when you think about this time, this was long before the DNA and the RNA have been identified as molecules that take genetic information. But still, you see, there are two different departments in this institute for experimental genetics and for human genetics. This was extremely modern, and equally modern was that they have had a department for neurochemistry and pharmacology. At that time, it was the opinion was that adrenaline and noradrenaline are transmitters that are important for blood vessels, but not for the brain. So it was not known that these transmitters have a specific role as neurotransmitters also for signal transduction in the brain. But one of the daughters of the Vogts, Marthe Vogt, she was a chemist, and she brought this research direction into power very early. There's also anatomy, histology, histopathology, physiology, psychology, even phonetics, and in addition, a photography and reproduction area, and the clinical examination ward, which had up to 42 beds for patients with neurological disorders. And again, it shows the very broad scope of this institute and the broad scope of the concept of the Vogts that really addressed the whole human being, the patient, while going back to the molecular, genetic, and physiological and anatomical properties that are contributing, of course, to a certain brain disease. So a very modern concept. They also understood that brain structure and function are not so easily connected in the human brain. In particular, physiological experiments are practically impossible. However, they collaborated with Ottfried Förster, who worked in Königsberg, which is now Kaliningrad. And Ottfried Förster was a very famous neurosurgeon who stimulated the brain of patients undergoing surgery for epilepsy. And by stimulating the brain in these patients, in order to better understand what is a functional representation of motor activity, in particular in these brains of patients, they also mapped the brain. and came up with one of the first homunculus, also long before Sherrington has published it, who was also a visitor in Königsberg. And they also introduced the approach of comparative anatomy and physiology, and compared physiological findings in different species with the anatomy of these species, and then interfered how the physiology of this different species would look like in a human brain. So indeed, a very comprehensive approach. And this is important to understand why we today emphasize very much the multimodal character of a human brain atlas. But let's further go. In addition to Oskar and Cecile Vogt, there was also Corbinian-Brodmann, and Corbinian-Brodmann analyzed the cytoarchitecture of the human brain, that is how the cells are being distributed. And you see here an example of a cytoarchitectonic image, and all the black cells, these are neurons that are arranged in a very specific way. And Vogt has published this very famous picture where on the left hand he shows the cytoarchitecture with the different cortical layers, and on the right hand he shows the myeloarchitecture, also with cortical layers that map to cytoarchitectonic layers, but nevertheless have a certain independence, and they are also named in a different way. Yeah, so you can map these two aspects to each other, but both aspects, they contain different types of information. And we know today from connectivity studies, for example, from tracing studies, that indeed the cells that are localized in a certain cortical layer, they have a specific connectivity. Again, this connectivity is different from brain area to area, but very roughly speaking, we can say that layer 4 is receiving inputs from the thalamus in particular. Layer 5 and 6 are mainly output layers. Layer 3 contains also large cells that are connected to other brain areas, for example, in the other hemispheres. So there are certain rules of the relationship of the cellular architecture and the myeloarchitecture that already the Vogts and Corbinian-Brodmann started to explore. However, when we think today about something like an atlas, we have more something like Google Maps, but for the brain and mind. So it's not a flat map, it's also not a sphere that we have in mind, but it's something that really reflects the different aspects of brain organization. And it also goes beyond the simple atlas in the sense that that this atlas does not only contain the outlines of different brain areas, but also information about the areas. What is the cell density? What is the myelo density? What is the connectivity of a certain area to other areas? And to bring all this together and also be able to get hands on this data and to use it, this will Timo explain in the second part of this talk. So we think something about Google Maps, but for the brain. But still, we are building our atlas concept of the Julich-Brain atlas on some principles that are very old. So we start from the cytoarchitecture because we feel this is really a very strong and reliable and also very popular reference for mapping. We consider also intersubject variability in our atlas, which is a new development as compared to Vogt's time, because we have to acknowledge that brains are very different in their cytoarchitecture and their anatomy. And this plays, of course, a role when we compare our maps with those of healthy human subjects, for example, of patients. We are convinced that it's important to go really down to the cellular level, and therefore we have introduced the BigBrain model. An atlas today should be multimodal, so we bring in fiber and molecular architecture and link it to cytoarchitecture. And of course, we have to be capable to compare these types of data with other data coming from functional imaging experiments or connectivity elements. And this requires to bring everything into one common reference space. So where we are starting with the Julich-Brain really is creating a comprehensive and coherent framework of the cytoarchitecture, the fiber architecture, and the receptor architecture as a reference for brain function. And within these modalities, we always precisely know what the area is. We always know for a receptor architectonic region, what is the cytoarchitectonic area that is behind. And we also know it for the fibers. So here we can really be very sure that we talk about one and the same area. Also, all these different methods have been acquired in different brains, which differ also between each other in their macro and in their micro anatomy. We start from the cytoarchitecture, and this is again another picture of cytoarchitecture coming from area 17. This is the primary visual cortex, and you see here the border to area 18. This is a secondary visual area, and without going into detail, it's immediately apparent that the cytoarchitecture is different when you compare the right-hand side of the image with the left-hand side. And in particular, on the right-hand side in area 17, you see a very broad layer 4, which has already which can also be subdivided into sublayers. And this layer 4 is so massive because it receives input from the retina via the lateral geniculate body. And this massive input is reflected by this very broad thickness of layer 4. And this is different in higher visual areas which do not have this direct massive input from the retina. So you see here how connectivity is reflected in the cytoarchitecture as well. Unfortunately, for most of the areas that we have in the human brain, gyri and sulci are only approximate predictors of microstructural areas. And this is an example from the primary auditory cortex, areas TE1, which are located at the Heschel gyrus. This is HG. And you see the very characteristic shape, the mushroom shape of the Heschel gyrus. And indeed, the primary auditory cortex occupies it. But if you look a little bit more closer, then the transition to area TE1, which is not, or TEI, which is not the primary auditory area, is not well defined by the fundus of the sulcus, and the border to TE2 is also, let's say, not precisely in the fundus. Maybe this is good enough, but if you further go to higher auditory areas, then you can see that there are borders between TE22 and 21, which are not, which cannot be predicted by any sulcus. They are located right at the wall. And if you look to STS2 and STS1, the red and the blue area, then again, their border is located in the wall. So some areas do have a rather strong link to sulci and gyri, and the primary auditory cortex would be such an example, but others do not. So in general, we have to say that sulcus-based maps are not good predictor for predicting, for identifying cyto or microstructural areas based on the sulcal pattern. And when we look to individual brains, then we can see that also rather, I would say, evolutionary old areas like 17 and 18, primary and secondary visual cortex, they are quite variable. And we could show here in this publication from 2000 that the shape and extent of area V1, primary, area V2, secondary visual cortex, is quite variable. And also the border between these two areas is not predicted by any sulcus. So again, we have a lot of variability, significant variability, but we also have we have areas that cannot be nicely predicted by gyri and SaltSci. So how to solve this situation? Our approach was to map 10 brains, as I have shown it here for the visual cortex. These are 3 other brains where we have mapped the frontal pole areas FP1 and FP2, and these 3 areas again they differ a bit, but then we register and align them to a common reference space, which here is the MNI reference space. And then we compute the superimposition of these areas, at the end 10 areas, not only 3 as in this example. And we compute so-called probabilistic maps that reflect for each position of the reference space what is the probability that a certain cytoarchitectonic area is present in this particular spatial localization. And you see here that for this particular area, there is a lot of blue and green regions. That means there is a rather low variability that this area is present, while red means there's a 100% overlap or a very high certainty that this area is found at this particular position in reference space. And the cytoarchitectonic work that we started more than 25 years ago revealed a very detailed parcellation of brain areas where we did not expect it, honestly. And this is an example of the human insula where 12 areas are now identified and even more are existing. And these areas occupy and parcellate the insula, and on a higher level we can group them into granular, disgranular, and agranular, disgranular clusters or areas, but the finer subdivision still holds true, and that means the insula is really highly parcellated. And sometimes the insula is also being seen as a brain in the brain because it has so many functions. And when you now have a look to modern atlases that are gyrus and cypress based, then you can see that these atlases, they do not show this fine-tuned parcellation. And in particular, in the insula, they are rather simple in the sense that they identify the whole insular cortex. But the insula has many different functions, from somatosensory to motor to language to cognitive function. And saying that the functional activation is within the insula is the same as you would say, I'm going to Asia to vacation, and you don't know, are you going to India, to north, to south? Are you going to China? Are you going to Japan? There are quite some differences, and it is important to be rather precise if you want to have a good roadmap of your trip to Asia. And a little bit the same is with the insula. Yeah? It's a whole continent, but many maps do not reflect it. In that respect, we were really happy that one of our former doctoral students, Julian, has mapped the insula in this way. And indeed, this mapping corresponds to different functions. And this is illustrated in this image here, where we can see that motor functions, attention, somatosensory function, or also gustation, they are mapped to different cytoarchitectonic areas. So we see a very clear mapping of functions that can be recorded in fMRI experiments, for example, two cytoarchitectonically defined areas, and we see a disadvantage if, so to say, all these different functions are subsumed just under the label insula. Yeah, so this does also the functional imaging community does not bring it to the next step if the anatomy is so rough. So we feel it's really important to be precise only in terms of anatomy. So when we started the Human Brain Project in 2013, we had a so-called BigBrain model that was at that time the highest resolution whole brain model, isotropically reconstructed with 7,404 sections. We introduced this reference in order to be even closer to the cellular architecture. And at that time, our Julich-Brain atlas included 77 brain regions, primarily cortical areas, primarily areas that are closely related to primary sensory and motor areas, not so much higher associative areas. So when we look today to the BigBrain model, and you will learn about it later during this course, it's still the highest resolution entire model. There are many, many downloads, and we have included many maps of cortical areas, nuclei and layers, and many annotated data sets that have been uploaded. So it's really a true resource for the community. When we look to Julich-Brain atlas, then we have now much more brain regions, whole brain coverage. These data are available according to FAIR principles, and there are different formats: volumetric, surface-based, interoperable, and Zebra, and you will hear about it later. So this is how it looked like a couple of months ago, but the principles are still the same. And we will show to you further examples during the course. It includes cortical areas, subcortical structures, as here, for example, the thalamus, probabilistic maps that can be used in order to predict tracts or to compute connectivity or to identify the relationship to functional parcellations. The BigBrain model is, so to say, a second pillar that I would like to mention here in this context. It's coming from a body donor, and this brain has been fixed in formalin, then embedded in paraffin, and then cut into 7,404 individual sections. And while sectioning the brains, we have obtained so-called block face images prior to sectioning in order to create undistorted or relatively undistorted references of the histological sections that were then subsequently stained for cell bodies. So when we just stack or create a stack of these histological sections and do a very first 3D reconstruction, then of course it looks very odd and not like a brain, but our work together with Montreal colleagues resulted in a 3D reconstruction which is really highly precise and has led to a 20 micrometer isotropic reconstruction of a single brain, which has approximately a size of 1 terabyte, original data set. And in the last years, we then parcellated or started to parcellate these maps in a detailed way. And since the spatial resolution is so big and there are so much data, we really were benefiting from the progress in deep learning and AI. And Christian Schiffer has developed a very nice tool to map sections and to scale up the mapping of human brain sections. So equally important to developing such high resolution tools is also to analyze these tools. And based on deep learning, this is now possible in a way that better scales up. And such kind of data are going into different applications. And one application is from our friend and colleague Victor Girza, who is a neuroscientist and theoretician in Marseille. He is using the BigBrain data as an input data to inform a model, the so-called virtual brain, about cellular properties that we have obtained in the BigBrain. And he then combines this data with electrophysiological and neuroimaging data from a particular patient here that is undergoing epilepsy surgery in order to develop a model that is predictive and that can help neurosurgeon to better define the places, the regions in the brain where they remove tissue in order to decrease or to eliminate seizures of these patients. And there was a large clinical trial in France that is now finished and where now the analysis is going on. And we hope, of course, very much that this combination of high resolution data, clinical data, and a theoretical model really leads to progress in brain medicine. This is another example of recent work, and Alexei is here, the owner of these very nice 3D maps of the human hypothalamus. And again, the human hypothalamus is also a kind of universe. It consists of very different nuclei that have a very specific connectivity and also a very specific function. And importantly, some of these nuclei are very closely related to the vegetative and to the hormonal system of the human brain. And to understand what is their cellular architecture, what is their localization is extremely important. And such nuclei are too small and their contrast is too low in order to see it in functional or even in structural high-resolution MR imaging. So we want to go further and really approach the cellular level of the human brain, and Timo will talk about it much more in detail. But the challenge is that when we want to do so, we are really facing very large data, also within a section, but also when we think about the entire human brain, that will then sum up with a few petabytes, and to store these data, to handle these data, and to analyze them is extremely important. Let me also add in my talk that, of course, behind cellular architecture, we are addressing the receptor or the molecular architecture of the human brain, and this is one example of the hippocampus. And you see here little regions of interest that have been labeled for different neurotransmitter receptors, for glutamate, GABA, and adrenaline. And all these transmitters, they have different receptor types, like the NMDA receptor. And the concentration of these receptor types is very different in different brain regions, but also different in different layers of the hippocampus here. And it contributes, of course, to the functionality of the hippocampus. And as you can see here on the left side, we know precisely where we are with respect to the cytoarchitectonic parcellation of the hippocampus. And this is highly relevant not only for hippocampal function in healthy human subjects, but also in patients and Alzheimer's. And in Alzheimer patients, for example, we see a very low density of cholinergic muscarinic receptors, and indeed cholinergic substances are being used in order to decrease the symptoms of Alzheimer patients. This shows you a more general view that also in other isocortical regions, receptor distribution is extremely different. And we see regional and laminar within an area differences. And when we combine different receptor types of different neurotransmitters and use this as complementary information, then we can come up, of course, with a very detailed understanding also of what the cytoarchitectonic relevance is and the function of these areas. And here's an example where we did it already. It is Broca's region that classically has been subdivided only into two areas, 44 and 45. And if we study the receptor architecture, we see that there's more than a dozen of areas. And this makes a lot of sense when we think about the functions that are behind language that go far beyond only two different functions. So we found that Broca's region consists of many areas, and now we are studying what is their relevance. And interestingly, we see that when areas are very similar regarding their molecular or cellular structure, they share also rather similar functions, as has been shown here in this collaboration with our colleagues in Leipzig. And we have here identified areas of the sentence comprehension networks, and indeed the similarity in this sentence comprehension network was closer than the similarity to neighboring areas which are also prefrontal, for example. So we can use this multimodal approach to approach also Wernicke's area, and this is an example of cytoarchitecture, imaging, receptor architecture, and genetic information. So the idea is really to bring in these different modalities and to understand what is the relationship between them. And we have identified in this temporal region, for example, a new area called STS2. I have shown it already earlier. And we see a correspondence of this new area that was not present in any historical map with a tractographic parcellation that was obtained in a diffusion imaging study in living human subjects. So there is a close relationship of cytoarchitecture and diffusion imaging, as we can see it here. And we see also a relationship of cytoarchitectonic areas with a functional parcellation, for example, as can be obtained via meta-analytic approaches. But this relationship is a more complex one, which is not surprising in my view. So it's there is not an easy one-to-one relationship that you can say cytoarchitectonic area X has a function Y. For most of the areas, this is not so easy to make such a comparison. But we see rather a pattern of certain functionally defined areas contributing to a network that is relying on a certain number of cytoarchitectonically defined areas. And when we discuss such complex functions like overt reading or music production or music perception, it is of course clear that this is not one simple function, but it's a combination of different aspects of brain function that is necessary in order to perceive music, for example. Yeah, so you cannot expect that if you address such high-level function that you identify a single area behind, while when we are with our cognitive description rather close to the areas, for example, when we speak about motion detection or face perception, then we are much closer to the cytoarchitectonic correlates of this particular function. So for this Wernicke region, we also started to analyze and to compare the correlates of cytoarchitectonic and receptor architectonic organization with the genetic underpinnings. And indeed, gene expression is an extremely valuable modality that should also be brought into connection and with cytoarchitectonic findings. And Sebastian Bludau will tomorrow probably show you more about it. But the idea was here really to see whether the similarities that we observe in cytoarchitectonic organization, whether they correspond to receptor architecture, and whether receptor architecture has a close link and correlation to the genetic architecture of this region. And this is a result of the study. So we have analyzed the primary motor and somatosensory cortex and higher unimodal areas, the auditory cortex and the visual cortex. So 4 different functional systems. And in these functional systems, we have studied the gene expression, and this gene expression data came from the EBRAINS connected to the atlas using a tool that the group has developed here with Sebastian, with Timo, with Thomas Mühleisen and others. And the gene expression shows a certain distribution of visual areas in pink of auditory areas in orange, of somatosensory in light green, and of motor in dark green. And the same distribution pattern can be found when we group areas just based on their receptor architecture of neurotransmitter binding sites. So the take-home message is such multimodal analysis reveals indeed very deep organizational principles within these functionally defined systems, motor, somatosensory, auditory, and visual areas. And we see how the receptor and how the genetic architecture is changing from the primary area towards higher areas, towards associative areas. Last but not least, I would like to emphasize that we are also studying connectivity at the microstructural level, and polarized light imaging is a method that Markus Axer and team in particular have developed in the past. It is based on unstained histological section with a thickness of about 50 or 60 micron. And in these unstained histological sections, we can obtain, using the birefringence properties of the myelin sheets surrounding axons, the direction of fiber tracts. And the result of such analysis is shown here. On the upper left, you see such a PLI image, and the different colors code the different directions of fibers, while black indicates that the fibers are going very steep. And you see here a lot of differences in fiber orientation down to the single cell level. And in collaboration with Roxana Koolmans from the Amsterdam University, we can then also counterstain the same section using different antibodies in order to have an even more detailed understanding of what is the axonal architecture in comparison to different cell types. And this is kind of bringing myelor and cytoarchitecture, I would say, to the 21st century and a little bit closes the loop of what Vogts have been done more than 100 years ago. So let me finish with this movie coming from Markus' team that shows you really the challenge that we are addressing when we address the human brain. It's really a challenge related to the multiscale character of the human brain, where with microscopical techniques we can go very much into detail, but losing a bit the background and the surrounding. While with entire or with whole brain methods, we can go into the whole human brain, but we cannot go into the detail. So polarized light imaging is a method that is located at the intersection of microscopical techniques and whole brain imaging techniques. We really can see single fiber bundles down to the axonal level. And when now moving to the cerebral cortex, we can see that the orientation and distribution is changing. So this is, I would say, what Vogt would have dreamed about to make this really 3D and going into such a detail of the fiber architecture of the human brain, as can be seen in this particular image. And the resolution is 1.8 micrometer here for every pixel, and this allows you to get such resolution as you can see here. But when we want to go to the cellular and subcellular level, then of course we have to go to the sub-micrometer level. However, these methods like 2-photon or also electron microscopy, they cannot be done for the whole human brain. That's simply impossible. But having an atlas concept that is multi-scale from its very nature, and this is what Timo will present also, this makes it possible to link the different modalities to each other. And having said this, I thank you for your attention, and would like to invite you to closer follow up with us at our website. And I will stop now. I would say if you have immediate questions that should not wait, then let's talk about it. And otherwise, I would hand over to Sebastian, to Timo. Sorry, Sebastian is tomorrow.
Hi, thanks, Katrin. That was wonderful. You showed the data from 3 human brains, which you said for the probabilistic map. Did you have MRI also for each of those 3 individuals?
I mean, I showed 3, but there are always 10 behind. So every area is based on 10 brains. Yes, we do have MRIs, magnetic resonance images, structural, with a resolution of about 1 millimeter. However, some of them are rather old. They are coming from the mid-90s. But yes, we do have them.
Thank you. And also the molecular maps which you showed for earlier brain, not the ones from the Allen data. Those were IHC data series or those were transcriptomic data?
The molecular data are our own data. They are coming from our own lab. This is Nicola Palomero in particular. The Allen brain data are gene expression data that we are capturing from their openly available atlas. And in der Tat, Sebastian has made a tool that allows to link the Seattle data with our data. It's gene expression data. In principle, such tool would also work for other resources. Yeah, for us, it was important to go beyond the own lab and to link also data that are many, many thousand kilometers apart from each other. So I would say, Timo, you should continue because many of the questions, I think, or some of them will be answered by your presentation.
Yeah, okay. Happy to take over. All right. So I'm Timo Dickscheid. I'm also working in the Institute of Katrin Amunts and leading a research group on big data analytics. So I'm a computer scientist. That is also why I comment more on the technical side. And in the presentation today, I will focus on how we make these atlases that Katrin just described available on the web and in the cloud and accessible to you. This is a picture of my research group here in Jülich, a fairly recent one. So many nice people involved in this. And I have to say that, you know, making this available in the web, of course, requires a lot to happen before that. So is a pipeline that we typically work on in our research team that is, of course, doing image analysis on the supercomputers. So we have a lot to do with AI and image analysis in this undertaking. And of course, this all relies on high quality data being available. And Katrin mentioned this, that there are many large data sets, microscopic data sets available, which form the basis of this atlas ecosystem that you show, and which are based on histological lab work that is really invaluable for these studies. So I will explain the last part here today, but just want to make you aware of all of these steps, of course, happening as a prerequisite to that. So in the last years, we have made this precious and comprehensive information of the atlas available on the web as a part of the European EBRAINS research infrastructure, which is openly accessible and will be further developed in the coming years for neuroscience research. So when you go to this page, ebrains.eu, you will find several categories of services there, and you can share data. You will find brain atlases. This is what we talk about today, and to a large part also the rest of the week. But there will also be services for modeling, simulation, computing, and also health research. So several categories of services there. We focus on brain atlases, and I will try to give you a more technical overview now how the scientific work that Katrin has presented actually goes into infrastructure. So when we look at the brain atlas from a technical perspective or from a more formal perspective, we always distinguish a couple of ingredients that we have to model, and this is of course a reference space that we need, which basically gives us a coordinate system. just as we use coordinate systems of the Earth. Then we have a set of maps that actually define which brain regions or which structures are located at different locations in this space. And then we are typically also dealing with some form of taxonomy that gives names to these regions and also explains their relationships very often as a hierarchical tree, as we see it here. And you already saw in Katrin's presentation that our approach to build an atlas goes far beyond the simplified picture here, in that we are mapping very comprehensively, and that we are mapping in different reference spaces and different resolutions, and also according to different image modalities. So we really try to provide a comprehensive, detailed picture of the brain from very different facets of brain organization. In EBRAINS, we have in the past years developed atlases of different species. There is an atlas of the rat brain, of the macaque brain. The human brain is what we focus on today, of course. And there is also a support for the for mouse brain atlas, which is based on the work of the Allen Institute. We have developed software tool suite, which makes these atlases accessible and interoperable. That will be the focus of what I present to you. We have also developed a set of tools that helps to anchor new data to the atlases. Thereby facilitating that we further extend the atlas ecosystem in the future and also allow users and the community to do so. And last but not least, of course, we have developed tools for neuroscientific analysis that make use of these atlases. And since there are so many applications to analyze data with on the basis of these atlases, what we provide in EBRAINS is a selection of such tools. The different atlases that we have implemented in EBRAINS have over the years grown and have matured, and Katrin has already shown you that the Julich-Brain cytoarchitectonic atlas, which is at the heart of the human atlas ecosystem, provides more than 200 structures meanwhile, and the same is also the case for the red atlas that we are building here. The real challenge in implementing a software ecosystem for this atlas that Katrin already outlined is its multiscale approach. So I just want to summarize this again. We not only have here a parcellation map at the millimeter scale, which is what you typically see when you download a parcellation, which gives you a labeling of different regions, but we also have variability covered with these probabilistic maps that Katrin mentioned, which are typically collected in 10 different brains for the cytoarchitectonic mapping. And from there, we have, of course, the underlying high resolution information. As you saw, these maps are based on delineations at the micrometer scale at the level of individual cells. And although the probabilistic maps are actually provided at the millimeter scale, when we aggregate them between different brains in 3D, we want to provide access to the underlying microscopy data, since that is where we have the precision and where we can do quantitative experiments and learn about quantitative aspects of these regions, such as cell densities and layer structures. So the idea of the atlas is really to go from the region down to the microstructural detail, in principle to the individual cell, and on all these scales have the possibility to access data and to work with it. At the same time, there are these different modalities. So this path across the spatial scales, which is shown here, refers to cytoarchitecture. But as Katrin has mentioned, we are also analyzing fiber architecture, receptors, connectivity, and function in this atlas. And of course, these other levels also need to be integrated into the concept. Yeah. So we have here links that for specific regions, we have connectivity coming from different modalities. Let's say from neuroimaging studies, yeah, we have receptor densities measured in different regions. So very diverse and broad collection of data, which is constantly growing. And that's a challenge to really to really implement this into a software framework that is easily accessible and does not require you to search information in different repositories, download it, and process each part of these data with different tools and using different ways. That is the challenge that we were facing, and what we will show you this week, and what I will also show as an overview already now, is a framework that provides you first of all an intuitive and ad hoc access to all this information through an online web viewer, so that you can, just by opening a browser, already see and explore this information visually and interactively. Yeah. But then, of course, if you want to perform studies with this, and in particular, if you want to exploit the big data that is under the hood, you will want to program and write programmatic workflows with this data to really exploit the large amount of data that is around. So we have also a Python interface that allows to process and work with all this information and analysis tools. And in order to have this accessible, as already mentioned briefly, there are tools which allow you to bring data in and link it with the atlases. My presentation will focus on the things here on the right now. So one driving aim in building such an online atlas nowadays, of course, is to make it sustainable and to follow the well-established FAIR principles of data sharing, yeah, so that that we can make sure our data is reusable, other people can find it, can access it, can work with it. That's at the heart of the design that we followed here, and therefore all the individual elements in the atlas that you will see are actually curated data sets which you can find on EBRAINS. Yeah, so everything that you touch in the atlas, from the reference template to the parcellation maps to the linked data to microscopic image sections, they are all data sets on EBRAINS with comprehensive metadata openly accessible in the system, even without the atlas viewer. Yeah. So everything we see here is actually data set, and you will learn more about this during the week. What the atlas system is doing is it brings all of this information into a common anatomical framework and makes it processable and explorable through the 3D context and through the 3D anatomy. And this building such a system is only possible by connecting infrastructure aspects. So we are relying here on curation services that help us to do professional sharing and description of the data. We have the EBRAINS knowledge graph, which is a comprehensive graph database to actually store the complex dependencies between the metadata of the different data sets and atlases in a professional system, and make it from there accessible for programming purposes. And then we have these atlas services, which actually give us an entry point and an integration of all this data in anatomical terms. And behind the scenes, we also rely on really on hardware, computers, and storage systems, which in the case of EBRAINS are provided by European supercomputing centers, which actually provide the basis of where all the framework is built onto. The data curation services is something that everyone in the world can use, so you can really go to ebrains.eu, share data, and actually request curation of your neuroscience data set there. You will then be in touch with a curation support person who helps structuring the data. There will be a quick check whether the data is suited for publication on EBRAINS. And after going through these steps, you will end up with a public data set that has a DOI that is also recommended by high-ranking journals to share your data. And that is what we use, and what we also suggest other people to use to share their neuroscience data. The system has under the hood a comprehensive metadata standard, OpenMinds, which has been developed with the community over the past years, and Liu Batzil will explain in much more detail on Thursday, I think, what this is all about. Our Atlas services are then really built into this system. Yeah, so we have these data services with a metadata database behind. We have the infrastructure with cloud storage. And the Atlas services actually store their foundation data on the cloud storage, the parcellations, the maps, and the templates. We have services running as virtual machines on the infrastructure. And then as a user, you actually access our front-end services like the Atlas Viewer, which gives you access to all of this. You also find installable software that you can use on your own computer, and you have possibilities to implement your own scripts and workflows through the Python client to use all of this. It is also possible to use the atlases in high-performance computing workflows, which is something we are doing, and that people who do simulations and AI are also more and more using. So the software framework that we will look at the coming days is called Zebra, and it basically implements these red boxes here, an online viewer and a Python client that you can use for scripting and workflows. This is what we will focus on. So we call the online viewer Zebra Explorer. That is a software that really allows you to quickly dive in, to explore the Atlas without much, with very little learning curve. And Sebastian and Kim will show you the viewer tomorrow in more detail. I will just give you some brief examples in the coming minutes. There's Zebra Python, which is let's say, a fully fledged programming framework, which is still easy to use. And we will look at this in detail on Wednesday together. And there's actually Zebra API, which is a programming API, an application programming interface on the web, which actually the viewer is using to connect to the functionalities in Zebra Python, but which you could also use to implement your own applications on top of this. The online viewer already provides you possibilities to go through the whole setting I just showed. Yeah? So you can find the atlases there, you can find the probabilistic maps, you can find linked data here. This is just a very quick run through, and you will do this in much more depth tomorrow. Yeah? And then you can zoom in and you can switch to the BigBrain, the microscopic model that Katrin Amunts has shown. Yeah? You can really just zoom in with a scroll wheel, go down to the high resolution. And there you also find sections and data at the full 1 micrometer resolution level, right at your fingertips. So what you have seen here, you can simply do this in the browser yourself, and you will do it tomorrow, I guess. So this is really an online system that already allows you to go through this, let's say, this framework that I have outlined previously. The question is, how are these integrations done? So how do we link these different data types with each other and these different spaces? And there are, of course, a few fundamental principles. If we're using different reference spaces, such as the BigBrain template, which is a single subject brain at 20 micrometer resolution, and let's say the MNI space, which is an average MRI at millimeter resolution, we can, of course, exploit the Julich-Brain cytoarchitectonic atlas, where we have the structures actually available in all these spaces. Yeah. So for BigBrain, we have high resolution, very detailed cytoarchitectonic mappings in this particular brain, which you can find in the BigBrain. But we also have the corresponding probabilistic maps in the MNI space. Yeah. So by selecting these brain regions, you already have a link between these reference spaces in terms of the anatomical structures. Yeah. The same holds for going from the volume to the surface by providing the same parcellations also in the free surface spaces. We provide a link between those worlds, so to say. There is a second path, of course, which is the more common and maybe the more generic one, which is based on spatial transformations. So, of course, we can use image registration procedures to find corresponding coordinates in the different spaces. If you have worked with image registration in human brains, you will know that this is never perfect. Yeah. So image registration going from one brain to the other computationally is always a very ill-posed process, and depending on which tool you use for image registrations, you get a different solution. So corresponding coordinates have also always been taken a grain of salt, so to say. But it is an important link, of course, between these reference spaces as well. We are exploiting here a set of very precise transformations between the reference spaces that have been computed by our friends at Neurospin in Paris, and are based on diffeomorphisms, which are constrained by sulcal patterns on the brain. So there is a regularization here of this registration process in terms of major sulcal patterns. So this is structurally quite well controlled what the registration is doing there. Yeah. So, and then we always have these two possibilities. If I have a coordinate in the MNI space, I may look up which cytoarchitectonic brain region there is, and then when switching to the big brain, I can select the same brain region, so I will see a similar location in the brain. But I could also use the coordinate and basically just warp the coordinate to its corresponding one. And typically, of course, this will take me also to the same brain region. These links, we can use them to anchor data to the atlases. So one way of anchoring data is to do that by brain regions. Yeah, Katrin has shown some examples where we have measurements of receptor densities, for example, where in the experiment it was very carefully, the tissue was very carefully located in the brain, so we know that the receptor densities have been measured exactly in a particular cytoarchitectonic brain regions, and then we can link this data set by the name of the brain region in the corresponding atlas to the atlas ecosystem. But that's not always available. Yeah, often also we have measurements coming from labs which are linked to coordinates in MNI space, for example, and then we can use these coordinates to link with the atlas. One example is if you connect to the microarray database of the Helmholtz-Institute, for example, these gene expression measurements, they are linked to MNI coordinates, and we can use the MNI coordinates then to compare to assign the data to brain regions and to coordinates in other reference spaces. And if we have image data, we can of course use image registration methods right away. Yeah, so if we have data from a histological experience, we need not necessarily go through a reference space, through another reference space, but we could work on registering this data set right away to the space that we are interested in. This is of course not always possible, and especially for very high resolution volumes of interest, we basically have to use a high resolution reference model such as the BigBrain to do the anchoring, because an MRI template space does not have the level of detail that would allow to anchor it based on image registration. But these are three fundamental ways of linking data. The system that we have built, the software system, Zebra, makes this distinction explicitly in the software. So we distinguish between spatial entities that are linked to a reference space, such as points, bounding boxes, or images that show certain structures. And on the other hand, brain regions, which are linked to a reference parcellation in anatomical terms. And between this, spatial world and the, let me call it anatomical or semantic world, there are of course links. So by mapping the brain regions in different spaces, we basically realize the brain regions in a particular reference space and can then connect them to spatial entities such as points and bounding boxes and so on. We can also go the other way around. If we have a point given, we can assign the point to brain regions by performing an anatomical assignment, and Zebra supports with this. From these informations, we can run spatial queries and semantic queries into the system to basically ask what's there. Yeah? Given a brain region, given a region of interest, given a coordinate, whatever, we can ask the system what's there. What do you know about this location in the brain? And this way we obtain what we call data features, which could be measurements from a broad set of modalities, as of which Katrin has shown many in her presentation earlier this morning. The Zebra tool suite basically has these data features always modeled as a link to an external data set, typically on EBRAINS, but you can use it right away to access the data itself. So we can from there fetch the underlying image data or fetch tabular features such as densities or fetch connectivity matrices. And these are then available in interoperable formats. So typically, if you pull image data through Zebra, it will be a nifty image. If you pull tabular features through Zebra from some online resources, it will typically have the form of an Excel sheet or in Python, it's a pandas data frame. Same holds for connectivity matrices. Yeah. So you basically go from a specification of a location down to a complex query of multimodal descriptions of these locations. We have seen that this can be done in the viewer, but tomorrow we will have a closer look at the Zebra Python library, which allows to do this really in terms of programming. Yeah, so we can here really write typically short Python code that finds brain parcellations, downloads them, searches certain regions. It can process views of the online viewer and extract information in these views, download full resolution data from the BigBrain, 1 micrometer data in certain regions of interest for you, and do all of this in a fully reproducible fashion. Yeah. So what this means in particular is that if you do a publication with such data, you can actually provide a code snippet with your paper, which reproduces how you got the data and makes this directly reproducible for readers of the paper and people who want to follow up with this work. This is Zebra Python, and tomorrow on Wednesday we will have 90 minutes to go through this in more detail together. I talked already about Zebra API, which you could use for application development. And we have also developed, which is quite interesting, an interface of the data-led software, which is, if you don't know data, it's kind of a Git for data. There's now an interface that you can actually use this to pull data right away from EBRAINS data sets. And that's quite interesting because if you find any data or information about the brain in Zebra, it will always give you access to the corresponding DOI and to the corresponding EBRAINS dataset ID, and you can use this right away to clone the data with data-led EBRAINS. So it's really straightforward to implement reproducible workflows with state-of-the-art technologies with this atlas and using these technologies here. I'm just checking my time. I think I can still take a few minutes. I want to make sure we also have some minutes for questions later on, so I try not to take all the time, but a few more minutes. So one thing that is particularly typical when you work with Zebra is to go from a location to information, and I would like to highlight this process a bit more here incrementally. So usually in the different Zebra interfaces, you start with some form of defining a location of interest. For example, you have a point in the M&I space, yeah, and then you can use Zebra to basically assign brain regions in a probabilistic fashion to this point using the probabilistic maps. So you would get such a list as you see it here with different brain regions and different probabilities, how likely it is that the point is actually inside these brain regions. From there, you can retrieve the probabilistic maps and all the information about these regions, of course, and you can use these to query for multimodal features that have been linked with these brain regions. This is one part. Often you don't have a specific point, but you might have an image with carrying some information. For example, an fMRI activation map that shows you some blobs of activation in the brain. And of course, you can also use such images and compare them with the probabilistic maps. Maybe do a cluster analysis to split them into different components, and then you can assign to brain regions by correlating these blobs with the probability maps. Basically giving you then another entry point into the same functionality of doing region-based feature queries. But if you have spatial information, as we see here, if I really have an image with a spatial structure on its own, I can then also use this directly to run spatial feature queries, just so to say, not going through the brain regions, but right away using the coordinate information to do additional feature queries. All of this is supported by the framework in a quite efficient implementation, and we will have a look at that on Wednesday. Let me just check time again. Yeah, maybe just one last thing. Katrin already showed you the differences in cytoarchitecture in the different brain regions. I'm not repeating that here and now, but as I motivated in the introduction of my presentation, the idea of the Atlas system is also to give you access to the quantitative information in the different brain regions. So if we look again here, as here we have an image patch from the visual system and one from the motor system. And we know that the cytoarchitecture is very different, but we are of course also interested to get quantitative measures of the cytoarchitecture in terms of cell densities and so on. And we have established a couple of data sets, which you will also see in more detail, I guess tomorrow, which actually measure layer-specific cell densities using precise cell detection. So here in our lab, we have developed a state-of-the-art cell segmentation tool with deep learning, which was very successful in a recent international challenge on cell segmentation, which directly estimates the contours of cell bodies. So it gives us a quite stable measurement of the cell instances, even if they are overlapping with their particular shapes. And we have used this algorithm on a significant range of brain regions in different image sections to quantify the densities of cells in the different layers. So you see here basically what's available in an already published data set, and we are working on a data set that is even provides measurements in a much denser sampling across the whole brain. I think we will get back to this also in the coming days, to this data set. And one last thing I want to mention is everything you see here is extensible. So the software that we have developed is separate from the content. Yeah. So the atlases, the parcellations, the data that you see is not built into the software, but the software is configured to show these contents. But we will constantly increase the amount of contents that you see there, and you can also contribute content yourself. One way to do so is simply to share your own data on EBRAINS and work with the curation team to link it to the atlases. It will then automatically pop up in the tools that we see here. We can also, you can also directly integrate data into Zebra if you're running your own installation, which is the case usually when you use the Python client. So independently of what we provide, you can then simply add your own data and work with it. And it is also possible to implement custom functionalities into the online viewer because it has a plugin system included, which is kind of like you can implement add-ons for your Firefox browser. It is also possible to implement add-ons for the online viewer. And some add-ons have been implemented, and I guess that probably Sebastian and Kim will show you at least the one for Jugex, for the gene expression analysis tomorrow, I guess. Okay, and with this I will probably end. So we have a couple of minutes also to take your questions and discuss a little bit with you. So I will stop my slides here, and happy to hear your questions.
Right, thank you, Timo. I have a very basic question. When you say anybody can share data, it could be in any modality?
Yes, so sharing data on EBRAINS is in principle you can do any modality. There is of course always a brief check, right? So we have to make sure, for example, if you have human in vivo data, we have to make sure that it takes the safe way and so on. Yeah, and if it's not related to neuroscience, it might be rejected. But in principle, you can share any modality. However, the level of integration with the software is of course highly dependent on the modality, right? So when I said that data sets shared on EBRAINS automatically pop up in the system, it is true in a sense that as soon as you link it to a brain region, Zebra will show you this data set if you ask what is available for the brain region, but it will not always be able to load the data in the data set, right? So for example, for our cell densities, Zebra knows these types of contents, and it can already show you the underlying data and allow you to work with it. If you share a data set with a very exotic modality, of course, Zebra cannot know how to load and process it. Yeah, it would still show it, but it would then probably just forward you to the EBRAINS data set and not actually interpret the underlying data. So long story short, you can in principle share any modality subject to ethical checks and so on, and privacy checks, but the level of support in the software varies, of course, in terms of what modalities, how exotic the modalities are.
And currently, this is all, whatever you mentioned, is to neurotypical brains, or it's also including the diseased brains?
We don't distinguish this in terms of data sharing, but the atlas that we're building here and the data that I've shown you has a clear focus on studying the healthy brain, right? So with the atlas, we want to describe the structure of the healthy brain, yeah. But in EBRAINS, it is also possible. So we don't make this distinction for the data sharing. Otherwise, I maybe just to outline this again. So now we have hopefully a bit better picture of what's there. The presentation tomorrow will focus on the Atlas viewer. So Sebastian and Kim, and there will also be Julia Tönnissen, will take you through the viewer and show you examples, and you can get your own hands on and do a few exercises, I would guess. Julia Tönnissen will be there to briefly show you how we anchor volumes of interest from histological experiments to the system and make them available in the viewer. So this will be all about working in the, visually guided workflows in the browser. On Wednesday, it will be me again, and I will show you Python notebooks. So we will do some programming. I will assume some basic Python knowledge there already. So if you have no knowledge in Python, you will still be able to follow and watch along, but it will probably be a little bit tricky. So we don't assume much, but we will really work on Python code. And on Thursday, it will be Liuba Zehl, who explains the metadata, the EBRAINS knowledge graph, the metadata standards, and how we actually make sure that that data sharing in EBRAINS, and with that also the content modeling of the atlas follows the FAIR principles of data sharing. That's the idea for the week.
Very good. So then I think thank you for today. Also for you being here in such large audience. We appreciate it very much. And let's continue tomorrow with more hands-on session, and then I would suppose that more questions come with during this course that we can hopefully answer.